Greetings, Pencrest biology students. Mr. Del Prado here to talk to you today about meiosis. Um, if you look at the slide in front of you, you can see that we actually have a comparison between mitosis and meiosis. These words are very similar, and the processes are similar to one another, but you will see that there are some key differences between mitosis and meiosis that are going to result in different cells being made when you compare the two processes together. So, big picture here, if you don't want to read through all of those little facts on the left-hand side, the big picture is this. In mitosis, your regular somatic body cells start out with 46 chromosomes, and then when they divide, you have two cells each with 46 chromosomes. In meiosis, this only happens in your sex cells. So if you think about this, your sex cells and someone else's sex cells might actually someday combine together. If you don't understand that, go talk to a health or phys ed teacher or go talk to mom and dad. But either way, at some point, your sex cells might combine with someone else's sex cells. So it should make sense that in those cells, sperm cells and egg cells, we need to have half the number of chromosomes that the regular body cells would have. So in the case of human beings, we have 46 chromosomes in our somatic cells. That's just our regular body cells. But in our sex cells, we have 23 chromosomes. Our sex cells are said to be haploid. They're also said to be 1N, which means that they just have one copy of each chromosome. You might remember that your somatic cells are 2N. They have two copies of each chromosome. So in order to get that, in order to get our chromosome number in half, meiosis is essentially going to have to divide that original cell twice so that the end result, if you look in the bottom right-hand corner, is going to be 4 cells, each with 23 chromosomes. Yes, I'm well aware that the 4 cells in the bottom right don't actually have 23. They kept it at just 2 for simplicity's sake. So I'll probably be going through a fair amount of vocabulary during this. Just to recall, some of these words you've actually had before. If you see like 1N or 2N, it's just the number of pairs of chromosomes. 1N is going to be one copy of each chromosome. 2N is going to be 2 copies of each chromosome. 1N equals haploid. 2N equals diploid. Okay? The zygote is the fertilized egg. That's when a sperm and egg actually join together. If you hear me use the word gamete, that just describes a sperm cell or an egg cell. Haploid cells, in humans' case, cells with 23 chromosomes. Fertilization is when the sperm cell and egg cell come together. And this one might make you giggle, but it's kind of important in biology. Sexual reproduction is reproduction between two individuals. Remember, there's lots of organisms out there that can reproduce asexually, and they are essentially producing clones of themselves. But in sexual reproduction, we're actually producing offspring that their DNA is actually a little bit different than mom and dad's. And so that's what this process is all about. Here's the big picture for meiosis. If you look at the first 4 cells here, 1, 2, 3, 4, you can see that essentially it looks very similar to mitosis. You have prophase, metaphase, anaphase, telophase. But because we're cutting the chromosome number in half, this cell is going to get split into 2 cells. Then these cells are going to divide again. So what we've done is we split up meiosis into 2 separate categories. Meiosis 1 consists of prophase 1, metaphase 1, anaphase 1, telophase 1. And then when these cells divide, we call it meiosis 2. Prophase 2, metaphase 2, anaphase 2, telophase 2. So yes, the end result of meiosis at the very end is going to end up being 4 sex cells. All right, so same thing as mitosis. Your cells are going to start out growing. That is interphase. The cell is actively growing. We have chromatin in there. We have the nucleus in there. And in fact, many cells spend the majority of their life in that stage. All right, here we go. So in prophase 1, very similar to prophase, the chromatin condenses into chromosomes. The nucleus starts to disappear. You can see the centrioles and the spindle fibers starting to appear there. But the big picture, or the big important piece of this, is this part, is that the homologous chromosomes right here, let's say this is from mom, this is from dad, this is from mom, this is from dad, they line up literally as a couple, all right? And this is kind of important because what's going to happen is in your sex cells, and this is amazing, these chromosomes, the homologous chromosomes, will actually touch and cross over one another. And the DNA from this chromosome and the DNA from this chromosome will actually get exchanged with one another. Same thing with these two homologous chromosomes. The end result is going to be a cell that has like jumbled up DNA, like or a chromosome that has some of your mom's DNA and some of your dad's DNA on it. This is called crossing over, and it is incredibly important in meiosis because it essentially increases the genetic diversity in a cell. Because now you have not just mom's DNA or dad's DNA, you actually have a mixture of your own unique DNA inside of your cell. So let's take a closer look at that. Okay, so this is it. This is going to be crossing over. There's my homologous chromosomes right there. Okay, and you can see with our sex cells, they'll line up next to each other, but look, this one chromosome is literally like crossing over and touching the other one. Okay, it's called a chiasma, and this is where these two chromosomes kind of like link together. But look at the end result. You end up getting two chromosomes. Well, this isn't really just mom's chromosome. This isn't really just dad's chromosome. They now have this unique DNA that essentially is uniquely your own. I'm showing this in two chromosomes, but this happens with all 22 pairs of your chromosomes, where we get this crossing over and this jumbling up of DNA. And that's super important to increase genetic diversity. That's why you're not really 50% mom and 50% dad. You actually have genes that are unique to yourself. And we get this from crossing over during meiosis 1, specifically in prophase 1. Okay, look at our chromosomes now. They have like little tips that actually have different colors on them because the DNA is scrambled up a bit. But metaphase 1, very similar to metaphase. You can see they're lined up. You can see the spindle fibers are attached to the chromosome. We're going to get ready and we're going to pull these apart. Now, this is the key piece. This is a key piece of the difference between mitosis and meiosis. Remember in anaphase and mitosis, the chromatids split apart? That is, like the X's got cut in half. That is not the case here. In this case, and this is where the cell is going from being diploid to being haploid, the chromatids aren't splitting apart. It's the whole chromosome is going to get moved from this side, and it's going to go over this way. This chromosome is going to go over this way. This one goes this way. This one goes this way. Okay? That ends up leaving us with a cell, if you think about it. We have a cell here with 4 chromosomes. If we cut this in half, each one of these cells will then have 2 chromosomes, half the number of the original cell. In your sex cells, it would go from 46 down to 23. In anaphase 1, the homologous chromosome, not the chromatid, the whole chromosome gets pulled to the opposite end of the cell. And there's another way in which we can increase genetic diversity, and that is this. This chromosome might go this way, and then this one would go this way. This chromosome goes this way. This chromosome goes this way. But this can also happen. This chromosome might go this way. This one might go this way. And then this one still might do the original, go this way and this way. If you think about that, it creates sex cells that are all a little bit different of one another. That's called independent assortment, because it's easy for me to say like, oh, well, this chromosome is going to go over to this side, this one's going to go over to this side, but that is not always the case. Sometimes this one will go this way, this one will go this way, maybe this one will go this way, this one will go the other way. They all assort independently. And so what you end up getting is a big jumble in these sex cells that have all sorts of different DNA, which is super important for genetic diversity within a species. Okay, here's telophase 1. In telophase 1, you can see the cells are dividing. Okay, what's going to happen is the nucleus is going to reappear. There's the centrioles. They should be disappearing because we don't need the centrioles and the spindle fibers anymore. And look what you end up having. You end up having 2 cells, okay, with half the number of chromosomes of the original cell. Okay, but now we need to split these chromatids apart. Okay, and this is called meiosis 2, when we take each of these cells and we split the chromosomes into chromatids. If it sounds like mitosis, it is. This cell essentially in meiosis 2, it's just going to go through mitosis, and so is this cell. The end result is going to end up being 4 cells, and they'll each have, remember, these are replicated chromosomes, they'll each have 4 chromatids in them. So here we go. Let's watch. Okay. Here's prophase 1, or excuse me, prophase 2. You can see there's the nucleus again. There's the spindle fibers. After, by the way, meiosis 1, the cell does not go back into interphase. It goes right into prophase 2. Okay. There's our spindle fibers. I'm presuming, let's take a look at metaphase. There they are. This is happening in each cell. The chromosomes are lining up in the middle of the cell. You can see the spindle fibers are pulling them away. Here's anaphase 2. Oh, look, the chromatids separated this time. Okay. Telophase 2, we essentially are going to end up getting then each cell with half the number of chromosomes, and now we have unreplicated chromosomes in there. So 1, 2, 3, 4. Okay. If they are sperm cells, you end up getting 4 sperm cells, each with 23 chromosomes. Okay. Ladies, for egg cells, what you end up getting is this uneven division of cells in meiosis, and then you get one gigantic egg cell that has all of the nutrients for a developing baby. Okay? Difference between meiosis 1 and meiosis 2. Meiosis 1, in anaphase 1, the whole chromosome moves to the opposite ends of the cell. In meiosis 2, we're splitting the chromatids apart. Meiosis 2 is essentially the same thing as mitosis. In the big picture, why do your sex cells do this? Because remember, your sex cells are the only cells in your body that might fuse with someone else's sex cells. They need to have half the number of chromosomes. Think about it. If you had 2 cells with 46 chromosomes, the baby would have 92 chromosomes. And we'll talk about in class why that cannot happen. So I hope this was understandable for you. I know that it's a lot of vocabulary, but we'll have some other accessories for you to help you understand the process of meiosis. So we'll see you on the other side, and thanks for listening.